Okay, so let's start. Thank you to all people that are connected, and welcome to this new Neuro'binar today. So a quick reminder that the Neuro'binar is a 1-hour web conference, and we have 3 presentations, 2 from PhD students that present the work of each other. And this presentation will be continued by question about PhD work. And after, we have a principal investigator. Today is Adam Williamson, and it's a 20-minute presentation about his research in Marseille, and it will continue by 15-minute question. So a quick presentation. So our speakers are Gaelle Alhaddad and Pedro Belio Mairal, and Adam Williamson is a principal investigator. So Gaelle, she has a master's degree in psychomotor therapy, and during her master's degree, she specialized in the evaluation and rehabilitation of French and Arabic handwriting. And she is now under the supervision of Dr. Marie Cloncan, and she's doing her 2nd year of PhD in the MULO team, which is Music, Language, and Writing at the Laboratory of Cognitive Neuroscience. And after Gaelle, we will have a presentation by Pedro Belio Mairal. So he also has a master's degree in translational medicine from the University of Barcelona. And his research is about the neuroglia interplay in synaptic dysfunction in the context of neurodegenerative disease. So he is now in the 3rd year of thesis under the supervision of Dr. Santiago Rivera and Emmanuel Nivet at the Institute of Neuropathophysiology. Gaelle, it's your turn.
Hello, everyone. I'm going to present Pedro's work at the Institute of Neurophysiopathology under the joint supervision of Dr. Rivera and Dr. Nivet. His presentation is titled MT5-MMP in Alzheimer's disease. Alzheimer's disease is a devastating neurodegenerative disease that is expected to increase exponentially, as you can see on the graph, and reach pandemic numbers in the next decade. Thus, there is an increasing interest in understanding the pathological mechanism of the disease and how to tackle them. Currently, we're starting to understand that the progression of the disease is based on the complex interplay of several factors that positively influence each other. Among the most prominent, we have the amyloid beta accumulation, neuroinflammation, and synaptic dysfunction. Ultimately, the progression of the disease leads to cognitive decline and eventually death. Unfortunately, there are no effective treatments available to stop or reverse it to this day, and in part, this is due to its complexity. Therefore, there is a strong need to understand the mechanisms behind Alzheimer's and identify reliable therapeutic targets. The widest accepted theory of the probable cause of Alzheimer is the so-called amyloid hypothesis. I'm just going to put my pointer, it's easier. This hypothesis claims that the accumulation of amyloid beta drives the onset and the progression of Alzheimer's disease, leading to neurofibrillary tangles, disturbed cell trafficking, neuroinflammation, and other complications. Amyloid beta is produced by the cleavage of APP by beta secretase that generates the C99 fragment, which is further cleaved by gamma secretase, giving as a result amyloid beta. But other potential treatments that were directed towards amyloid beta have yielded disappointing results in clinical trials. However, recent discoveries point at C99 as a potential new target that correlates better with the symptomatology of the disease. So within this specific context, Dr. Rivera's group has shown during recent years that metalloproteinase called MT5-MMP might have an important role in Alzheimer's disease. Dr. Rivera's group has demonstrated that the lack of MT5-MMP in a mouse model of Alzheimer's disease was preventing the accumulation of C99, the formation of amyloid plaque as well, and the levels of inflammatory cytokines. Moreover, this resulted in preserved synaptic plasticity and cognitive performance. Further work in Dr. Nivet's group confirmed that these results could be replicated in neurons generated from human pluripotent stem cells obtained from patients. So overall, this extensive work elucidates the potential of MT5-MMP manipulation to hinder Alzheimer's pathological processes. As a result, they are now interested in acquiring a deeper understanding on the pathological mechanism of MT5-MMP. So, to dig deeper into MT5-MMP, it is a transmembrane proteinase involved in the processing of several proteins, and it has diverse physiological function. It is formed by an extracellular domain and a C-terminal part. The extracellular region is composed by a signal peptide and a propeptide that precedes the catalytic domain, linked to a hemopexin region. And this C-terminal part is composed of a transmembrane and an intracellular domain. Knowing this, they generated several constructs with modified version of this protein to identify those domains that are responsible for its pathological functions. Interestingly enough, they found that only the deletion of the transmembrane and or the intracellular domains was leading to the reduction of C99 and the production of amyloid beta. In other terms, this means that the detrimental functions of MT5-MMP in the context of Alzheimer's disease may be due to non-catalytic interactions that influence the processing of C99. Consequently, now Pedro wants to decipher what are the exact regions of the C-terminal part that is responsible for the MT5-MMP pathological role. So regarding his future plans, and based on the comparison with MT1-MMP, another metalloproteinase with opposite results when deleting the intracellular domain, they defined amino acid groups that could be implicated in the interaction with C99. Then they generated a series of constructs with deletion of those selected groups or substitution with alanine residues. Furthermore, they generated two chimeras carrying either transmembrane or intracellular domains of MT5 and MT1. So far, Pedro has found that specific modification and defined amino acid groups are not sufficient to modify the processing of C99 in human immortalized cells. Nevertheless, he has identified different roles of the intracellular and the transmembrane domains, where the intracellular domain seems to be involved in the transport of C99 towards gamma secretase cleavage, whereas the transmembrane domain is implicated in the stabilization of C99 and in blocking its processing or degradation. Finally, they generated diverse adeno-associated viruses carrying these key modifications to translate their work on human neurons with Alzheimer's phenotypes derived from pluripotent stem cells. Ultimately, Pedro hopes to define the roles of these two domains with the objective of manipulating them to modulate C99. He expects to demonstrate that this can have a positive impact in the physiology of human neurons, preventing synaptic dysfunction. So finally, all the work Pedro has done wouldn't be possible without the help of his colleagues, and he wants to make sure that I remark how much he appreciates the efforts that they make. Thank you.
So hello, we are here so that today I can present Gaelle's latest experiments on the effects of Biscriptuality on graphomotor dynamics. Gaelle is in her second year of her PhD, and she's conducting her thesis under the supervision of Marie Loncamp within the music, language, and writing team in the Laboratoire de Neurosciences Cognitive. So usually whenever we bring up handwriting, we almost certainly have this image of cursive handwriting in mind. We tend to forget that the alphabet that we write is the product of thousands of years of a script evolution. And that Latin, hundreds of different alphabets exist, and wait, because I'm going to change. Okay, sorry. And like Latin, hundreds of different alphabets exist and are a result of evolution. So even though around us, Latin is the main script that we use, There are in fact other types of scripts that are as used and in other parts of the world. However, the most common combinations remain between alphabetic scripts. For Gaelle's thesis, the two alphabetical scripts that we will be studying are the Latin and the Arabic scripts. In Latin and Arabic scripts are widespread both separately, but are also combined in some countries that here are presented in red, where I'm pointing. In these countries, people are required to learn to speak and to write both the Latin and the Arabic alphabet. These people are called Biscriptuals because they are experts in two different writing systems. It is interesting to compare these alphabets because they are opposite in terms of writing direction and sense of rotation. French is written from left to right and in anti-clockwise rotations, and Arabic is written from right to left in clockwise rotations. Mastering both of these scripts thus could involve very different constraints on the graphomotor system because of their opposite characteristics. And that's why it is important to study Biscriptuality, also because it is becoming more and more common with migrational movements and globalization. Very little is known about the behavioral organization and occurring consequences, whether they are positive or negative, when writing two different graphical systems. And to this day, there are no published fundamental researches on French-Arabic Biscriptuality. Thus, there is no information on Biscriptual brain organization. Try to write a word or trace a graphic pattern, and you will notice that it's highly rhythmic movement. Similarly, when you write on a graphical tablet, you will get a lot of information and variables of the dynamics of the trace. If you trace loops, for example, we can analyze the variations of n of x and y as a function of time. And we can see that the trace is highly oscillatory and rhythmic. And it's the frequency of the trace and the differences in oscillation coordination that will determine the stability of the trace patterns that we can see through the roundness of the loops and their degree of slant. Based on that I've exposed regarding the Biscriptuality and the stability of graphomotor patterns, the first question that we ask ourselves is, are there any difference between monoscriptuals and biscriptuals in terms of writing coordination dynamics? So from the one side, we have people who know only one alphabet, and on the other side, you have people who write two alphabets, who will also frequently change opposite writing directions and sign of rotations. So if there are differences, we can ask ourselves if they could be compared to the bilingual advantage, where bilinguals are more efficient than monolinguals in switching languages, or the bilingual cost, where bilinguals show interferences from one language on the other. Will the biscriptuals be disadvantaged compared to the monoscriptuals? In this case, the patterns will be more variable and less coordinated, and they will be slowlier, or on the contrary, they will be more advantaged, and we will observe more stability in graphomotor behavior in terms of coordination and speed. To verify this hypothesis, a study was conducted. So a group of right-handed French monoscriptuals and Arabic biscriptuals participated in this study. The biscriptuals had all learned Arabic and French since their childhood and were experts in both alphabets. The task consisted on 4 conditions. The participants traced loops in different writing directions and senses of rotations. Variables indicating the degree on coordination and the stability, as well as spontaneous tracing of the loops between scriptuals and monoscriptuals were there verified. Sorry. Oh, no. So basically, the main results from Gaelle show that there are a lot of interesting results in terms of writing preferences. However, the most significant result that she wanted me to expose is that the effect on group tracing frequency. Biscriptuals have a higher tracing frequency than monoscriptuals and trace about a half loop more per second, as you can see in this graph. This could indicate that biscriptuals could have an advantage in terms of writing speed and coordination compared to monoscriptuals. And this is the result that Gaelle decided to study further for the rest of her thesis. Based on these results, what one could wonder if this increased efficiency comes from, and what could explain this different behavior in Biscriptuals. So better efficient of the motor system because the more intensive and varied motor training, or it is whether because of a better executive functioning based on the literature of bilingualism. So it is a consequence of constantly switching between scripts and handling differences from one alphabet onto another. To be able to search deeper into these two underlying mechanisms, in her next study, Gaelle will investigate the cerebral activity associated with the same loop tracing task in Biscriptuals and monoscriptuals that we saw before, with EEG that is synchronized with the graphic tablet, as you can see here. With EEG, we will be able to evaluate the power of the cerebral oscillations indexing motor control. In parallel, she will evaluate through behavioral testing the fine motor skills and the executive functions of Biscriptuals and monoscriptuals and correlate to their handwriting's coordination stability. And that's it. Thank you very much for your attention. And if you have any questions, you can ask Gaelle.
Okay, thank you, Gaelle. Thank you, Pedro. So if people have any question, you can click on raise the hand, lever la main, or write your question in question and response. Gaelle, just before people ask question, I have a question. I see on your results that you have, even if Biscript people that are Biscriptual, they are better in one direction than the other. They seems better in one direction than the other. Is it because they are more expert in one language compared to the other? How do you explain that?
We argued the preference for tracing from left to right, which is close to the French writing direction. First, because they are right-handed, and that we know that when we as right-handers trace, it is easier for us to trace with our hand that is getting away from our body trunk. So it's easier to have opening movements. It could also be to the fact that our Biscriptuals were actually people who were expatriated, so they live in France. So for the time being, they were more exposed to French rather than Arabic, even though they learned it for more than 25 years because they lived in an Arabic country. The third hypothesis that we have is that it is easier to trace French. So this is why, because they found it easier to use this language, they would develop this preference towards this specific tracing direction.
Okay, thank you. So we have two questions, one for Gaelle and one for Pedro. So the question for Pedro, so it's from Pascal. Yeah. So I think, Pascal, you can speak.
Can you hear me?
Yeah.
Right.
Yeah.
I don't know why I cannot activate my camera, but nevertheless, I will ask my question. Thanks very much for both of you for your presentation. I have a question for Pedro. I was wondering, I would like to come back to the activity of the C-terminal domain. I was wondering if other metalloproteases other than MT5 have a high level of conservation in this domain, and do you think this activity you identify is specific for these metalloproteases?
Thank you for the question. So far, we can talk about MT5 and MT1. MT5 and MT1 are two metalloproteinases from the same family that they are highly similar. Their amino acid sequence is very similar. But the main difference that you can find in these two, between these two proteins are specifically in this intracellular domain. Okay. So basically, yes, we think that the function that is showing MT5 in the terms of modulating C99 levels, it's specific for MT5 because also we have checked what happens if you delete the intracellular domain of MT1, and basically you have the total opposite behavior. So when you delete the intracellular domain of MT5, you have a decrease in the levels of C99, whether when you delete the intracellular domain of MT1, you have a huge increase of C99 levels. So yeah, I mean, yes, we think that the function of the intracellular domain of MT5 is quite specific for this protein.
Thank you very much. Really interesting.
Thank you.
Okay, we have questions for Gaelle, one on the discussion and two from the question and response. Did you, so the first is higher frequency tracing always better, and are there differences in accuracy?
We considered that the higher frequency in our case is an indication of advantage, because we had also measured variation of stability, and we saw that there were no differences between monoscriptural and biscripturals in terms of stability. So that meant that even though there was an increase in frequency, there was not a detrimental effect on the stability of handwriting. So they had more speed without having the loops that were more degraded than others. So yeah, no differences in accuracy and stability. And for the left and right-handed, we did not compare it with biscripturals or monoscripturals, but I had already done a little project on that where I compared left-handed monoscriptural French monoscripturals and right-handed French monoscripturals, and there were no differences in terms of stability of handwriting, nor in terms of tracing frequency.
Okay, you have a question on the discussion, which is regarding the Biscriptual paradigm. Why did you choose a simple loop sequence and not actual sentences or words?
Okay, loop tracing is used a lot in graphomotor handwriting, graphomotor and handwriting studies. We chose specifically loops because we were using the graphomotor approach to coordination, and since we had to calculate the PR, so we needed ellipses to be able to do that. So we had to use either words who contained letters such as L and E, or loops to be there. And since we had biscripturals, so people who wrote in Arabic and in French, we could not also present Arabic writing to the monoscripturals. So we thought that proposing loops was the best trade-off, as we could manipulate the writing direction and the sense of orientation that would perfectly replicate those two scripts. It is also a test that showed to be very sensitive towards these differences, and so that's why we intend on keeping it for further research. Do I have another question? No.
I think there are a couple of people who raise their hand in the chat, could be, or
I can't see that.
Ah, okay.
Brilliant. Is Pierre talking to us? He's muted.
I think that it's your turn. I think Theo said that there is no time for more questions, so we should move to your part, Adam.
Okay, he is unmuted now.
Okay, so, sorry, I was speaking and my micro was turned off. So we have no more questions. So Pedro, you have a question on the question and response. So you can write your response to Santiago Rivera. And now it's the time of Adam. So we have Adam Williamson today. So Adam holds a master's degree in science in electrical engineering at Texas Tech University in United States. And he received his PhD in mechanical engineering in Germany, where his research was focused on the integration of nanostructure into silicon photodiode. And from 2011 and 2014, he was a postdoc in the same university in Germany. And in 2014, Adam joined the Institute of Neuroscience, Institut de Neuroscience des Systems in Marseille. And he is a recipient of an ERC starting grant in 2016 and also an ERC proof of concept grant in 2020. And his research now is focused on in vivo application for numerous new electronic devices and brain stimulation methods in pathophysiological neural network related to epilepsy. So thank you, Adam. It's your turn.
Brilliant. Okay, I will share my screen. And let me just say, Gaelle and Pedro, that was fantastic. I know if I watched or had to watch someone present my slides, I would throw my computer. So excellent work, both of you. Really, seriously, it was very good. And I will start a timer. I have 20 minutes, and I tend to talk too long. So with that, and that, and that. Okay, you can see my screen. Yes, everything's good.
Yeah, it's perfect. It's
Brilliant.
perfect.
All right, so today we are talking about the application of something called temporally interfering electric fields, and we will specifically be talking about it in epilepsy. I was asked to give a very quick introduction about where I am situated. So I am a PI here at INS, at the Institute of Systems Neuroscience. I guarantee these numbers down at the bottom are not correct anymore, but let's say that there's approximately 120 people here. Somebody might say 140, but there's a lot of us. There are 4 teams. It's a UMR, so it's mixed. So with about 40 plus permanent members, you have people from AMU, people from INSERM, people from CNRS. For example, me personally, I am permanent with INSERM. We have all of the 5th floor in the red wing of the Faculty of Medicine, but recently took over the blue wing as well. And so my group and all of our new labs are in these absolutely beautiful facilities that have been renovated here on the 5th floor in the blue wing. So we are at Timon. As you can see, this is, I mean, the campus is beautiful. They really can't say anything negative about this. So the outline of the talk today will introduce temporal interference. And then I think at the end of that section, everyone should be at least very competent to understand the next couple of sections. The next section will be laser TI, which is where we basically revisit the previous section, but we do it in a super cool way using lasers. So, and I believe this was let's say the topic of why it was most exciting or why I was invited to give this talk. But then I want to end with something translational. I want to show that this is not just useful in tiny little rodents, that we can actually scale this and we can scale this to people. And then I'll give, I think, some contextual conclusion. All right, so let's start off with temporally interfering electric fields. Whenever I give the talk or a similar talk, I like to start with this slide, and then I like to say that this is the only slide in today's presentation that is not mine. This belongs to a very good friend of mine now. His name is Nir Grossman. He coined the term temporal interference. And so basically what we're looking at is a method of brain stimulation that uses 2 frequencies instead of 1. On the left-hand side of the slide, we see some blue electrodes at a frequency F. And on the right-hand side, we see a frequency that is in black, that is also F, but it has a slight offset. Now the frequency F on the left in blue is very high. It's a radio frequency. It would be like kilohertz, like 2000 hertz, something that we know does nothing to brain and to neurons. And this is then replicated with the black. So we have 2000 hertz again, but that slight offset, that delta F, that is an interesting frequency for brain stimulation. So here in the example, let's just say that it's 5 hertz. So now we have 2000 hertz and we have 2005 hertz. And at some interesting point, geometrically located as a function of the spatial orientation of those electrodes, we have, and can be very deep below the cortex, we have constructive and destructive interference. And my group and Nir's group and 1 or 2 other groups now have shown that that envelope stimulates neurons. So it's a very, very cool way to begin to create extraordinarily deep stimulation, but using topical electrodes. Now, we click to the first results that we published. This is a series of not just cool looking pictures that are supposed to represent mouse brains. These are full electrodynamic models of the mouse brain, including all sorts of subcortical structures and complex permittivity of tissue that we have in my group. We are highlighting only one side of the hippocampus just for illustration for today. Now, on the left in A, you see two electrodes that are going to be the blue and black, let's say, from the previous slide that are giving those two stimulation frequencies, but the references are off in the chest, which was previously a fine way of creating some transcranial stimulation in mice previously. And this is what they did in that original 2017 paper. What we demonstrated is that although effective to cause some individual cell firing somewhere in the hippocampus, it's really terrible at creating higher level focalities that can evoke really interesting network events, particularly interesting for epilepsy. So we started orienting these in little pairs, and we created lots of little pairs, as illustrated in B and in the right side of B. So parallel and perpendicular to the axons in the hippocampus. Lots of more information is possible on this. Just know that when you're orienting these things preferentially to axons, it's better. That's what we'll just say. That was the takeaway message of what we looked at here. So let's investigate what happens when we create one of these focal spots down here because of the overlap of those electric fields in the hippocampus from our transcranial stimulation. Sure enough, if you look at some stimulations, depending on the orientation, and it doesn't matter, we still target the same structure, we're just oriented a little bit differently. And we see we have this electric field that is down in the hippocampus, located at the portion of the hippocampus that we wanted to stimulate. We can make envelopes out of sine waves, and something that was very unique to our group is we've shown that you can make these envelopes out of all kinds of other waves. Specifically shown in this publication were square waves. And there are some arguments that show how transmitted power is actually significantly more effective with the square wave TI. But we won't, again, that's a little bit more electrical engineering. We'll avoid that. We want to just talk about how cool this is as a transcranial DBS. So if we look at a video, we can see this video is obviously slowed down. We're looking at two frequencies. We're making some TI in our little mouse here at 1,200 hertz and 1,250 hertz. That's giving us a 50 hertz envelope. So you can see the time scale there with 50 hertz is going to be 20 milliseconds. So we're coming up and down. And this works the same way if we looked at the other orientation. So it's the same. We have the same spot, and we have the electric field increasing and decreasing as a function of that envelope, but it's very deep. So we would be creating an excitatory stimulus in the case of the hippocampus and trying to evoke some type of epileptiform activity. The most interesting slide, I always say when I give this presentation, is this slide, because here you can see that the difference frequency is 0. We have 1,250 hertz and 1,250 hertz, which means this video, although slowed down, we can't see that because it is flickering at 1,250 hertz, right? So there's no envelope and therefore no stimulation. It's just two radio frequencies that are doing nothing. The field does in fact look like that, but none of the neurons are firing. Now, a lot of you are doing rodent experiments, so I put this slide in just to say, generally, we have surgery, we implant our mice with these little pins, or they're little transcranial pins or electrodes. We leave them a few days, depending on the protocol, to recover from surgery, and then you can have really any number of stimulation days, and it's all locked to video footage and freely moving, and works exceptionally well in all sorts of other freely moving applications. Obviously, you could do it anesthetized, but we did all of this previous work freely moving. So the results of the initial study were, in my opinion, absolutely fantastic. And let's specifically look at A and B, because these will be these preferential orientations. In A, we took one of these mice, or we took a group of mice, and we implanted a stimulator, a traditional twisted pair stimulator, and we evoked seizure-like events, these focal seizures, the big focal seizures to the hippocampus. We see, okay, we have our stimulation, and then about 10, 15 seconds later, we have these enormous seizure-like events. But that's been known for decades and decades that you can do exactly this. If you have this 50 hertz stimulation from a wire implanted in a specific portion of the hippocampus, you will have these seizures. What has never been done before, which is what we did, is in B. We have a transcranial stimulation that creates the very first non-invasive DBS. It's a focal stimulation inside of the hippocampus, but there's no electrode in the hippocampus for the stimulation. We create identical, and I'll go into detail in a moment, identical seizure-like events that are indistinguishable from their implantable brothers. Now, looking at the C and D, we simply say that doesn't work because those orientations are non-preferential. I would love to get into this, and depending on the amount of time that's remaining in the talk, I will definitely get into the non-preferential orientations and how they can also be extremely useful. But for now, let's just stay with A and B, and we'll say, okay, we can make little mice. We can have a non-invasive TI here, as we see in B. We can compare that to these implantable electrodes. We have seizures that, let's say, in these two panels, A and B at the bottom, they look the same, but they're also behaviorally the same. So it's indistinguishable from the phases of the Racine scale, a scale used to qualify and quantify the quality of the seizure that the animal's having, the severity of the seizure. The two forms, the non-invasive and invasive, are making the exact same seizures. If you give this to a blind reviewer, they're unable to distinguish what is TI and what was an implantable seizure. And again, with durations and RMS voltages, everything looks exactly the same. So it really is the first completely non-invasive DBS for excitatory stimulation. And that's something we would find very interesting in seizure localization. So the laser TI, let's go back and we're going to make these seizures, but we're going to introduce some of these very, very cool organic devices that we have. So we copy the first picture again that Neil Grossman had created in 2017, but we introduced the idea of light, right? So we're going to take some transducers. We're going to take these specific organic transducers, and for today, it's absolutely not important that anyone care or know about N-type and P-type materials and this organic material called P.PSS. We're just going to call this a transducer, and all of this tiny little organic device, what it does is if you illuminate it with deep red light, let's say 660 nanometers, it will convert that red light into an electrical stimulation of the same frequency, which is what we've attempted to illustrate here. You illuminate the device and it creates its complementary electrical signal on the cortex. Excuse me, so we're using this as a transcranial but subcutaneous stimulation. So here we have these implanted on our little mouse, and we want to create, again, that same type of envelope stimulation down in the hippocampus. And so here is illustrated, I think, extraordinarily well on our three-dimensional brain structure. Again, we see we have these subcutaneous light transducers, and we're going to illuminate, and we see that even in simulations initially, we see, okay, this is going to work very well with that orientation, and with this layout, we'll have, as we see here in one, a very nice envelope that will be down in the hippocampus, and up in the cortex, right up next to that light transducer, we're going to have only that carrier frequency. We'll have no envelope, and so as we discussed earlier, we'll have no stimulation. So experimentally, once this is implemented in mice, it's exactly as the simulation predicted. We have a phenomenal looking envelope inside of the hippocampus, and we have almost nothing, arguably even smaller than is illustrated here, let's say amplitude modulation in the cortex. And we don't have any let's say, we don't have any electrophysiological activity evoked in the cortex either. So here's what little mice look like. You have a surgery, you can implant these on the cortex, you can put them directly on the skull. If you wanted to, you could use these light transducers on top of the skin, but that sort of defeats the purpose of using the deep red light. Namely, that we put these below the skin, we put the skin back, and then we can illuminate the skin directly, and then that's how the stimulation takes place. So the little mouse can run around and you can play laser tag. And again, these interictal-like events, whether we're doing it with the laser TI or the direct stimulation, they are indistinguishable from one another, both in the amplitudes and durations, and then also into the frequency ranges of those interictal-like events. We have really, really, really nice beta gamma oscillations that are created when we stimulate that specific region of the hippocampus with those specific frequencies. So that ends the laser TI and sort of an introduction into the idea of making envelopes from topical electrodes that can stimulate deep. But the question might come, and it has come to me in a couple of reviews, that's great, Adam, that's great. You could do that in these little runs, but did I not see the Bujaki-Barini paper telling everyone in detail how transcranial stimulation doesn't work? I said, yes, I have seen that paper, and it's also not relevant to what we're doing because we're not doing transcranial stimulation. We're doing temporal interference, which is using a completely different set of frequencies which penetrate skull and tissue completely differently. So let's get into that argument. Here, we will initially look at some mice, and we're going to scale this to not mice in a moment. We take our mice, we're going to have a high frequency control, meaning a 130 hertz stimulus. And you'll say, why did Adam say high frequency? Because classically in electrophysiology, anything over 100 hertz to 200 hertz, clinicians refer to as high frequency stimulation. So no problem, we're going to have this control. And they get to stimulate the hippocampus, and we're going to be fair, we're going to give them the closest pair of electrodes to the hippocampus. Then we're going to take our TI, we're going to have a TI high frequency stimulation. So we're going to have two radio frequencies that will create a 130 hertz envelope way down in the hippocampus. We take our mice, we make our mice epileptic. The idea is we have these epileptic mice. This is not to indicate that the treatment is sham, HFS-TI, and HFS. We have different groups of mice. So some mice will receive no treatment, some groups of mice will receive the high frequency TI, and some mice will receive the actual high frequency, just this transcranial high frequency. Now, in the epileptic mice, we have lots and lots of these nasty, nasty, nasty interictal spikes. If you look at someone's very nice publications like Fabrice Wendling, the way he is categorizing these interictal spikes is based on amplitude, duration, rate, and area of one of these spikes. And what's fantastic is if you look at the TI HFS as a therapeutic DBS that is completely non-invasive, it reduces the spike. It reduces it by a lot. And more importantly, it reduces the character of those interictal spikes. It in fact crushes those interictal spikes such that it's very difficult to even identify them because the area, duration, and amplitude of those spikes is so small. Now, I will say something nice about transcranial stimulation because I do like it. I don't want to say that it's not working. I think it's very, very nice. I think these are complementary methods. In the case of fast ripples, so pathological fast ripples, we did not see any significant decrease between the two methods. There was a decrease for the stimulation methods compared to the sham, the unstimulated mice, but both are doing this complementarily. And we know that because cortex is also involved in these fast ripples. So that makes sense. That's a very nice result. So again, fantastic. In mice, we have a completely non-invasive therapeutic DBS for our epileptic mice. What can we do with that? Well, we can remember we have our little mouse, we have our focality in the hippocampus, and we were told that the Bujaki-Barini paper has told us that this doesn't scale. And in their paper, they used cadavers. So I made cadavers. We made many, many cadavers. We took cadavers and we stimulated them and targeted their hippocampus. And what we showed is that with our method, if we wanted to create one of those 130 hertz therapeutic envelopes, specifically in this example to the anterior portion of the hippocampus, we can be really focal. We can be very, very focal. This is, in fact, a, let's say, completely non-invasive DBS, unfortunately, in this specific example, on a cadaver, but, spoilers, it's coming. So let's look at this for a moment. We take one of those SEEG electrodes and we see, okay, very nice. If we're on one of those shallow contacts, we have a higher overall amplitude, but we have very, very little modulation. And as we move down the electrodes of that SEEG that we've put into this cadaver, we have more and more amplitude modulation. You can see it comes all the way to 0. And again, that works with sine waves or square waves. And if we were to double the amplitude for the traditional transcranial stimulation, we to the amplitude modulation that is attainable with the TI method. Okay, so that's excellent. That worked out. And it happens to work out in people as well. We have full simulations, electrodynamic simulations using Sim4Life, using SEEG implanted patients, and let's say compared to our SEEG implanted cadavers. And if we apply this therapeutic non-invasive DBS, just like in those little mice, we see a complete reduction in those interictal spikes, those nasty interictal spikes. And again, as an anecdotal picture, but I mean, it really is the situation. It becomes super difficult, even for my very talented students, to identify the spikes anymore because the spikes start to have this character compared to this very nasty traditional interictal spike character. And just as in the mice, these area, duration, and amplitudes of these interictal spikes are absolutely, absolutely crushed. So where are we going from here? The idea is focality. If I were to click back up, this is focal, but it's maybe not as focal as a DBS. This simulation accounts for some tissue propagation. It's not in the other hemisphere, but it's like the whole hippocampus basically looks like that. Okay. So is there something we can do to increase focality? And so what we did, and we're very proud of this, being the first people to do this, was why take 2 frequencies when we could take 4, 6, 8, 10, 12? Don't add just the frequencies, add the envelopes. So we take an envelope from 2 frequencies and an envelope from the other 2 frequencies. We add those together and we create super huge envelopes. Then you can reduce the overall stimulation applied from the individual pairs, collapsing this envelope amplitude back to the original, which was already effective. And we're going to, by definition, increase the focality. Saying this one more time, in 3 dimensions with a cadaver, We had old TI, which was fantastic. It's below the skull, it's below the cortex. It's definitely subcortical, but I would have a hard time arguing with some people in my institute to tell them that that's like focal, right? Well, with this method of multifocal TI, what we have now coined MTI, you can see here, this is the topic of our patent, is we have focality as good or better than implantable DBS, but we use topical electrodes and we add those various envelopes together to create it. So where are we going with this in general? The idea is n to the number n. You could have 2 frequencies, as we have done very effectively already. We could have 4 frequencies, as I illustrated in the, let's say, 2 slides previously. And then we go to essentially an infinite, I mean, we wouldn't use an infinite number of electrodes. That's silly, because already with 8 electrodes, 8 pairs, so 4 envelopes, we have this fantastic DBS style focality. And what's great is this allows us to implement multiple focalities at the same, so you can stimulate multiple places at the same time. And we have now dynamic control of these. So we can steer these different focalities around the brain, whether it's inside some of these initial preliminary mice, whether we're doing that inside of cadavers. And so we hope to have some ethics approvals to begin to use this so-called MTI in our patients. So I am right at 19 minutes. I will just say some concluding marks and we go directly to the questions. The concluding marks are, as you would expect in epilepsy, we use SEEG to stimulate to find our seizure foci. So as you can imagine, if you had these little mobile focality stimulation focalities that we could move in and around other parts of the brain, which were not previously implantable due to things like eloquent cortex, that would be fantastically interesting. We would complement the SEEGs very, very well. And if we were exploring resective surgery operations, we would have improved delineation with that, but we would also have the ability to test lesioning, test to see if certain regions, when we inhibitorily shut them down, as we've shown we can do, if that would be a great spot to cut out the seizure focus, if that would be a great resective target. So we can do that. And I just want to comment, every engineering friend I know that does anything with an electrode immediately says it's a DBS. I don't have any problem using this as a long-term implant with a DBS that would be subcutaneous, but it's like not the most interesting thing that I find right now. I think that this type of work for exploratory activities in epilepsy is really, really what I want to do with this method. So in conclusion, I will just say, I've got the 20 minutes, so I won't say much about this. I will just say this is a phenomenal method with the virtual brain, with Victor Gier's virtual brain. We combine our electrodynamic models, we combine it with the network models to make this patient-specific TI. And you could imagine at some point, you don't need any of the implantable electrodes. We simply connect you to what looked like a previous set of EEG electrodes. We're able to perform all the DBS and recording and ideally therapeutically intervene in that person's epilepsy. So that's hopefully the future of INS using TI. And let me just say thank you then to everyone. So the team, 2 postdocs, Dong Hak Byun, Malin Yejbi in Sweden, and then PhD students are Florian Missy, Emma Acerbo, Ibrahima Engom, and Boris Botanovsky. These 4 students are absolutely fantastic. I couldn't do anything without them. Funding is the ERCs you heard mentioned, EIC stuff. We're in France, everybody has ANRs. And our collaborators, VB Tech, super important. This is the startup company we have here at INS. Very, very close relationship with eBrains, part of the Human Brain Project. For me personally, in the US, excellent collaboration with Emory in Atlanta and with Feinstein, specifically with William Haynes and with Kevin Tracy. Karlinska, Peter Olofsson, Linköping University, Anders Friedberger, my very best friend in the world, Eric Gavatsky at SciTech, and then ITIS for all of those fantastic simulations we looked at, and obviously Imperial for my very good friend, Nir Grossman. So thank you very much. It looks like it's 21 minutes and 50 seconds. So I apologize for being very, very slightly over, but I am finished and could take questions if we have time.
Okay, thank you, Adam. It was a very, very great presentation. So yes, you have question. So the first question is from Rochelle Ackerley, and she thank you, and she asked if this could be applied to stimulating the periphery. She means the human nerves, and how focal can the stimulation area be here where the nerve is small and superficial?
So let's do the first question. That is a very, very good observation. I highly recommend that you send me an email and we have a Zoom, because I have so many awesome peripheral nerve results that are associated with this. It is, it's good for CNS. It's maybe better for PNS. It's Fantastic. So I would be very happy to share those results and discuss it with you. Regarding how focal. So I did this before. Everybody wants me to quote a number, right? But you have to modify your definition of focality based on the target. So if you have a group of neurons that are subcortical and you say they require 88 volts per meter, right? Depending on the number of envelopes we combine, you would say, okay, we combine in your application in this specific nucleus X, we'll combine 6 pairs of electrodes and we have an envelope that we locate, the dead center of that envelope is at that nucleus, right? All of these electric fields will then decay off towards those other 6 pairs. So then the challenge is just issued. You say, my nucleus has a width of Y, and I cannot have stimulation outside of Y because other neurons will have that have a threshold to fire are related to that specific electric field. Meaning, if you had these neurons that have 100 volts per meter, we put 100 volts per meter focality of electric field there, and then we continue to add pairs such that the distribution of that 100 volts per meter is narrow enough to accommodate your specific nucleus. So now, instead of talking so vaguely, I give fixed numbers. You could get 0.5 millimeter with as few as 10 pairs of electrodes in the human head, as we've seen. So that means if you wanted to nail something that was 500 microns and we were allowed to put 10 EEG electrodes, that's 5 frequencies that are, so 5 envelopes, yeah, that would be enough to have 500 experimentally in cadavers. This is going to be dependent a little bit on the size of the person. If they have an enormous head, maybe not, but just an average size head, average size cadaver, we have seen anecdotally that we can get sub 1 millimeter with 10 electrodes, altogether 10 electrodes. So five stimulations.
Okay, another question, Adam. Can your TI method be used for children patients with epilepsy? And do you plan to adapt it in the future?
So absolutely, totally, fantastically applicable to vulnerable targets or vulnerable populations, right? So you would say children is one. Why do we not prototype a lot of things in children? Because we all want to have successful science and successful grants. And if you put children in your ethics proposal, good luck getting that approved, right? It's a disaster. But this absolutely completely non-invasive stimulation, as opposed to something that has had some degree of efficacy, like an implantable intracranial electrode, like a DBS in say childhood depression or something like this. But why would you implant a child with a DBS? It's a disaster, right? This would do it. This you could do, whether you were doing some short-term intermittent therapy, you could just simply apply this EEG style electrodes to the child, target a specific region. Let's say that it was therapy. We have a project, we have a DARPA project that's starting that uses this in PTSD. So we're using some TI of these PTSD patients. It would work complementarily for the child. In fact, if you wanted to have that kind of focality, you need fewer electrodes because their head is smaller. So I think this is a fantastic method, specifically if we want to categorize children as a vulnerable population, where you say, there's lots of good invasive methods. Resective surgery is a fantastic thing. Intracranial electrodes are a fantastic thing, but it's very, very difficult to start doing that to children. So TI is a fantastic method for children.
Okay, you have a question from Boris Burl about your methodology, which is how do you decide the position of electrodes, intensity and frequency? And do you have a software or an open source software?
2. So initially we started using a finite element model method called with a commercially available simulator called COMSOL, but you can also do it with FDTD simulations from open source softwares like MEEP available through MIT, right? But those softwares are all relatively limited by the complex permittivity models of tissue that are available for those specific software packages. So now absolutely everything we do is through Sim4Life. So if you're interested in Sim4Life, obviously I have several licenses, permanent licenses for Sim4Life, and I'm happy to answer any detailed questions about complex permittivity, because basically what you're doing in the case of a simulation is you discretize the head and tissue into a mesh, and then everything gets a complex epsilon. It all gets an imaginary number, right? And then you use wave equations from Maxwell. You take the 4 Maxwell equations and you turn them into the 2 wave equations, and you propagate electric fields through the mesh, and everything just continuously gets attenuated by complex permittivity values. In the case of Sim4Life, they also take into account things like fiber and the activity of fibers. So those simulations can be significantly heavier and are, I think, scientifically significantly more interesting, which is why we've switched to Sim for Life. And that is but that's a whole presentation in itself, but I'd be super happy to talk to you in it offline or whatever we call that now, where you can come visit us and we can show you how all the simulations work. But Sim for Life is really, 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 I highly recommend that. Meaning you can calculate the structure of interest that you want and then an arrangement of topical electrodes that are available. For example, some arrangements are not possible with SEEG patients. They have bolts sticking out of their head, so you can't just put the electrodes anywhere. And you might not be able to shave all of the person's head, perhaps, if they're not interested. I think that they should be. And then we just calculate in advance where to put those electrodes to put the focality with the desired width in advance using Sim for Life. Sorry, it was a long answer. I hope that was clear.
Okay. I have just one question, and after it's finished, Adam, it's a very naive question, but in the brain there is, so I'm not expert on stimulation, but in the brain there is excitatory and inhibitory neuron. Is there a way with your stimulation to recruit more one type of neuron or the other, or is it impossible?
We have to log out of this meeting with everybody, and then we have to have a conversation, and I can show you the topic of a new patent we're filing. And so the answer is yes. The answer is yes. There is we have a method, we believe, that is very effective at targeting specific regions, but applying a differently modulated TI that affects interneurons differently than, say, excitatory pyramidal neurons. It's very, very interesting. It's very, very interesting. We play with the membrane capacitances with the modulation. So we're hoping that this will, as you said, preferentially target things like interneurons or excitatory neurons, if that's what was the case. But yes, there is. But I will be, Victor Jerza will shoot me if I explain it right now. But I'd be happy to talk to you after offline, show you how it works.
Okay, the last question, and after I will inform about the next Neuro'Binar, is from Christelle Bonnet. So related to your last answer is that you need to adapt the parameter to the target. And can you go as deep as the subthalamic nucleus?
I know Christelle's asking because she's interested in, has numerous publications for DBS in Parkinson's. So yes, you can definitely target the subthalamic nucleus. The reason that we haven't done any work there is because there's so much effective work with the implantable stimulator itself. So yeah, I had hoped to. So I briefly mentioned that when we were showing some of the cadaver experiments, talking about the use of replacing a, let's say, real DBS in a traditional neurodegenerative disease, right? So you can put the electrode arrangement subcutaneously. You could have a number of those electrodes, and you could reach the subthalamic nucleus effectively. But there's not a lot of great papers that indicate that the 2 electrode sticks that go into the head in patients, that's somehow more devastating, or like the replacing it's when you see the SEEGs, there's a lot of SEEGs, and we have a lot of tissue to explore, and the seizure focus is everywhere. In the case of her experiments, her expertise there, if you would like to try a non-invasive version of this, we know we can hit it, and that's fine. I hesitate to advertise it as a DBS because there's already so much excellent work that says, hey, you take 2 sticks, you put them into this specific region, and the stimulation is not, in that specific case, used as completely therapeutic reversal. It's not like you get a DBS and then you reverse the symptoms with Parkinson's or Alzheimer's or something like this. Those degeneration processes, there are many, many, many other experts that will tell you about the progress of that disease. You, to some degree, mitigate symptoms, but it's not a therapeutic cure. Whereas here in epilepsy, we're hoping that we use it as exploratory to let's say, enhance the cure. You can have a refined resective surgery. So yeah, absolutely, we do that as DBS, but I have not yet advertised it as such, because I think arrogantly in the past we've had organic this and organic that, and we've said anytime an engineer makes something, they immediately have previously said, oh, it's a new DBS, but it's not. There's fantastic data for traditional DBS. So
okay, thank you, Adam. I think we are on time. It's 2 p.m. 2 p.m. 02. So thank you for everybody to be here, and just to inform you that the next Neuro'Binar will take place on 22nd of March, so in 1 month specifically.
Pierre, I would say,
your...
oh, sorry. Yeah. I was just going to say, if I gave an incorrect, like an answer that was not acceptable, or if someone wanted more information, please just send me an email and I'll immediately contact you, and I can show you anything you want. Sorry, I didn't mean to interrupt you. Go ahead, sir.
Thank you, everybody. See you to the next Neuro'Binar.